Everybody have one? Now, we're going to try chapter 16 for a while on the top right. Chapter 16 is on our natural body defenses. And I say it this way. We have three lines of defense against infection, our natural defenses. The first line of defense is mechanical and chemical barriers. This is the first of our three lines of natural defense against infection. For example, a mechanical barrier is like skin, mechanically keeping germs out of our system, skin. A chemical barrier is like stomach acid, a chemical barrier. Now, sometimes a pathogen gets past those kinds of things. The second line must activate to take care of the infection. The second line I call PIC. Now, PIC is four things work together as our second line of defense. The P stands for phagocytosis. So now white blood cells start to engulf the pathogen, take care of it. The I stands for inflammation. We're going to talk about today two main examples of inflammation. One is the area around the cut turns red, inflammation. Also extra mucus in the nose, stuffy nose. So extra mucus in the nose or the cut turning red around the cut, that's inflammation. Now the C of PIC stands for complement proteins. Complement proteins. That may be a new term for you, so here's a definition, if you will, for complement proteins. These are blood proteins that do certain things to fight infection. These are blood proteins, sometimes called plasma proteins, that do certain things to fight infection. What they mainly do, by the way, these proteins down here help the first two processes to occur, mainly. And we'll discuss today some more details, but they mainly help the first two processes up there to go ahead and happen. Now the K stands for kinins. The definition of kinins is the same thing. This is a different group of blood proteins that do certain things to fight infection. This is a different group of blood proteins that do certain things to fight infection. Once again, what kinins tend to do is allow or help the first two processes to occur. The bottom two are protein types, help the top two processes go ahead and happen. So these four things work together as our second line of natural defense against infection. Sometimes those four things kick in, activate, but they cannot control the infection. Then the third line activates, take care of it. The third line is called the immune system. So if the first two lines cannot control the infection, the third line activates, take care of it. Make a note later on that the immune system is made up of millions of white blood cells called lymphocytes. It's also made of millions of antibodies. So those two things, by the millions, make up the immune system. It kicks in only if the first two lines cannot control the infection. Now, basically, the immune system is the next chapter, it's the next test. We're going to focus on, for today, the first two lines, chapter 16. So now some more details about that first line. Mechanical and chemical barriers. Again, a good mechanical barrier to infection is skin. The first two lines cannot control the infection. Sometimes they can. Now, the skin, if it's intact, is a great barrier to keep things out. Of course, the problem is cuts and surgery wounds and so on. That's a problem. Also called a mechanical barrier is mucous membranes. Think about the lining of our nasal cavity. That tends to trap things here to protect our lungs. And it's no fun to have a stuffy nose, but the advantage is trap things better here, protect our lungs. If they stay up here, then pneumonia shouldn't happen. Doesn't always work. Sometimes, despite the extra mucus, gets down there and does cause pneumonia anyway. A chemical barrier would be stomach acid, HCl. The pH of stomach acid ranges from 1.5 to 3. It's very acidic. So most bacteria die quickly in stomach acid. Here's another chemical barrier. It's an enzyme called lysozyme. Lysozyme. To be exact, this enzyme right here called lysozyme dissolves something called peptidoglycan. There's that word again. Lysozyme dissolves peptidoglycan. So what does lysozyme kill best? Gram-positives. This kills very well gram-positive bacteria in our system. Not so much gram-negatives. So lysozyme dissolves peptidoglycan, makes them fall apart, the gram-positives especially. There are 3 places we all have lysozyme. 3 places. In some of our white blood cells, we have lysozyme. Kill bacteria that is engulfed. Some white blood cells. Also our sweat, our perspiration, has lysozyme, kill bacteria. Also our tears have lysozyme. So most bacteria that fall on our eye die from the lysozyme in the tears. So some white blood cells and perspiration and tears. Now some more details for a while about the second line called PIC. Beginning with phagocytosis, under my job from the lab, there are 3 kinds of white blood cells that do phagocytosis. To begin with, neutrophils. This white blood cell type certainly does phagocytosis. What do they engulf? They engulf bacteria only. Not so much viruses, but bacteria. Then the largest kind of white blood cell, or a macrophage, they engulf both bacteria and viruses. Bacteria and viruses. Then the so-called eosinophils, they sometimes engulf the entire antigen-antibody complex. Just get rid of it. They engulf the entire antigen-antibody complex. So those 3 cell types do phagocytosis. Let's try to explain this. What are the 4 steps of phagocytosis? How does this work inside of our system? The first step of phagocytosis is called chemotaxis. Now this word means a chemical attraction. A chemical attraction. The question might be, how can a neutrophil find a bacterium in our system? It's a chemical attraction. It seems to work like this. Here's a large neutrophil And by comparison, here's a much smaller bacterium. That's actually too big for scale, but I'll call it that way. Now, once bacteria get in our system, they activate certain proteins like this. So make a note, C star means complement protein activated. And K star means kinins activated. The reason why they're activated is because there are bacteria coming into our system. They basically wind up coating or surrounding the bacterium. And it seems to work this way. If a neutrophil bumps into an activated protein like these, then the neutrophil moves automatically in that direction. Now here's the idea. If they're moving towards these proteins here, it's also moving towards the bacterium it needs to engulf. So the proteins create a chemical attraction which draws the neutrophil towards the bacterium. So as I've drawn it, the neutrophil would move from right to left as I've drawn it. Now step 2, attachment. Basically, the proteins go away to be reused somewhere else. And now the neutrophils can move over. I'm going to go this way to make it easier. Can move over and attach to the bacterium like this. So in step 2, they attach to the bacterium. But I want you to realize that there are 2 things a bacterium could have that blocks this step, and therefore saves the bacterium. One's called a capsule. It blocks that step. Also something called the M protein. So those 2 things can block this step and therefore prevent phagocytosis and save the bacterium. Let's assume they don't have those things, and step 2 did happen. They did attach like that. Now step 3. Ingestion. To ingest, the neutrophil has its plasma membrane change shape. It's kind of like this. The neutrophil's plasma membrane changes shape and sends out what I call these finger-like projections. That are called pseudopods. The neutrophil's plasma membrane changes shape, sends out pseudopods, and pseudo means false, and pod means feet. It's like false fingers. And pretty soon, the pseudopods fuse together. And now the bacteriums get ingested. I'll move it over. Here's the bacterium, drawn too big. Here's the plasma membrane material. Out here, the plasma membrane goes back to normal pretty quickly. So there is the ingested bacterium. Surrounding it is some plasma membrane material used to ingest. Now the last step is digestion. This involves structures called lysosomes. And make a note that basically a lysosome is a bag of digestive enzymes. A bag of enzymes. I'll draw some in, too big. These are lysosomes with my dots being digestive enzymes. So now a lysosome moves down and fuses there and basically dumps its enzyme into that compartment, its enzymes. Now the enzymes do both. They kill and digest the bacteria. They kill and digest. So pretty soon, this compartment has the waste products left over. that cannot be digested anymore. Those are waste products. So enzymes kill and digest here. Now, the compartment that contains the waste products left over is called the residual body. That's a compartment that contains the leftover waste products after digestion. Now, the problem is the neutrophil could possibly fill up with these waste products. It'd be a problem. So this happens. The residual body fuses with the plasma membrane. There's a very slight rupture of the plasma membrane, and the waste products are expelled. They do not want to fill up with waste products, so the residual body fuses with the plasma membrane, a very slight rupture there, waste products are expelled. So they find them by chemical attraction. Then they attach to them. Now they use pseudopods to ingest. They use lysosomes to kill and digest. They get rid of the waste products with the residual body. Do you think a neutrophil can ingest more than one bacterium at a time? Yeah. I do one, they can do several at the same time. Very efficient. Now some more details about inflammation on the handout. Inflammation. This is the eye, or pick. There are two main examples I want to mention again. One is the area around the cut turns red. That's inflammation. Also extra mucus in the nose, the stuffy nose is inflammation. I want you all to know 3 reasons why we bother to do inflammation. 3 reasons to do this. One reason is to confine the pathogen. Confine the pathogen. Secondly, to destroy the pathogen. Destroy the pathogen. And then finally, to repair damage. To repair damage. So why do we bother with inflammation? To confine the pathogen, destroy the pathogen, repair damage. Now I'm going to work in the details of inflammation. Now, as we talk about the last discussion question, the very bottom. Look at the very bottom. We're going to talk about complement proteins and what they do, some more details, and kinase. As we do this, I'm going to work in details and information, like how the cut turns red, and why do we have mucus in our nose sometimes, when that happens. So we're going to do a list here. Here are 3 functions of the complement proteins. 3 functions. I'm going to say it this way. They help out phagocytosis by doing something called opsonization. These are 3 functions of complement proteins. And make a note to yourself that opsonization is a fancy way of saying this. It means coating or surrounding. So complement proteins will take a bacterium in our system and they coat it. They surround it. Like this. That surrounding or coating process is called opsonization. And why do they do that? To chemically attract things to move in like neutrophils. That will cause a neutrophil to move in that direction to engulf the bacteria. They're helping phagocytosis by drawing in neutrophils. Then how do complement proteins help inflammation? For that, they stimulate 2 kinds of cells in action. They're called mast cells and basophils. So how do complement proteins help inflammation? By stimulating mast cells and basophils. To explain that, we're going to try some diagrams, kind of visualize some things, how they work. Here we're going to say that we have bacteria enter a cut on your finger, what you say. The bacteria will stimulate in that area the complement proteins in the area around the cut. Then the complement proteins in turn will stimulate in that area two kinds of cells. Mast cells and basophils. Bacteria enter the cut. They stimulate in the area around the cut those complement proteins, and they in turn stimulate mast cells and basophils. When these two kinds of cells are stimulated, I'm going to back up. Make a note, some on the side maybe. The mast cells are in tissue lining. They're tissue lining. But basophils are white blood cells in blood. If these two kinds of cells are stimulated, they fall apart, release their contents. And they are full of a substance called histamine. They release histamine. Now in my case, I didn't give enough room for myself. I'm going to go up the top, you keep on going down. Histamine will then stimulate what happened in the area around the cut. What's called vasodilation and capillary leakage. Histamine now makes those two things happen in the area around the cut. The first one means that blood vessels dilate, have more blood, not going out the cut, but around the cut, more blood. Now, somewhere in your notes, do a quick diagram here on capillary leakage. Looks like this. Here's a capillary, a microscopic blood vessel. This has walls that are one cell layer thick, and there are gaps between cells, like this. Here's a red blood cell that passed through single file. Here's a larger white blood cell that barely makes it. Now, normally, the red blood cells and white blood cells cannot leave capillaries. They can't squeeze out. However, histamine can affect the capillary walls. All of a sudden, they can squeeze out, like this. They can leave capillaries. So this is called capillary leakage. Histamine helps that happen. So the bottom line is this. In the area around the cut, we have more red blood cells than usual and more white blood cells. Not going out the cut, but around the cut. So what is the advantage of doing all of this? When bacteria enter the cut, what's going to happen? The extra white blood cells will engulf them as they come in. That's the idea. This all happens up here to make sure we have extra white blood cells in the area around the cut. They can engulf bacteria as they enter the cut. Most of the time, the cut should not be infected then. Sometimes, if you have too many bacteria coming in, the cut's infected anyway. But the idea is to attack bacteria as they enter the cut with extra white blood cells. Now, sometimes this happens. We have, let's say, viruses enter the respiratory tract. Let's say common cold viruses. Unfortunately, you have common cold viruses in the respiratory tract. Now, the viruses stimulate into action complement proteins. This is lining of our nasal cavity now. Now, they in turn stimulate mast cells. We have a bunch of mast cells in the lining of our nasal cavity. Now, they fall apart and release their contents, histamine. Now, histamine, what it does, depends on where it is. In the cut situation, it does these things up here. But in the nasal cavity, it does this, as we've all experienced before. It stimulates excess mucus production, the stuffy nose. Some folks take antihistamine to try that up. This is also inflammation here. When you wind up with a stuffy nose, with the idea being, once again, to trap the viruses better here. Protect our lungs down here. That's the idea. So by stimulating these two kinds of cells, the common proteins help inflammation to occur. Now, finally, I want to mention this. How can complement proteins help out the immune system? Now, I draw a line there because the immune system is third line, kind of a crossover. Now, they sometimes cross over and help the immune system by doing something called cytolysis. So sometimes complement proteins help the immune system by doing cytolysis. Now, cytolysis means make a bacterium, in this case, burst. So sometimes complement proteins help make bacteria burst. So here's the idea. In our system, we have two bacteria that are around. Only one of them has been neutralized or taken care of by antibodies. I'll draw it this way. These are two round bacteria in our system. One is free to cause us more problems. One's taken care of by antibodies. Now, in both situations, complement proteins get involved. In both situations. Over here, all they can do is this. They coat or surround the bacterium. Opsonization. To chemically attract neutrophils, perhaps. But over here is different. If there are antibodies there, made by the immune system, and a certain complement protein fits between them, up at C, complement protein fits there and binds, then you have a chain reaction, activating more proteins. Then you have this. These are seeds on the bacterium cell wall. These are activated complement proteins on the cell wall now. As they fuse together, they create a hole that goes through the cell wall and plasma membrane. So this binding activates a lot of complement proteins on the cell wall in a circle now. As they fuse together, they create a hole that goes through the cell wall and plasma membrane. So here, the cytoplasm becomes gushing out, the bacterium bursts. So if there are antibodies present, then they can make the bacterium burst. Now, on your test, if I choose this question, now I don't want these diagrams. I want these three things listed with a few more details I mentioned in the review. Not the diagrams so much at all, but these listed, plus a few details about those. Not the diagrams. Now, to finish up the new notes, we're going to talk about kinins. This is still a discussion question down at the bottom. Kinins have two functions. You can again say it this way in your answer. How can kinins help phagocytosis? Well, it's the same thing. It's called opsonization. They also coat or surround bacteria like this. So we're going to mention there that they help phagocytosis by doing opsonization, the coating thing. Then, how can kinins help inflammation? Inflammation. It's the same two things from earlier. They cause to happen vasodilation and capillary leakage again. So how do kinins help inflammation? By causing to happen both vasodilation and capillary leakage. So both histamine and kinins do these two things. To make sure in the area around the gut we have extra white blood cells to engulf bacteria as they enter the gut. So again, if I choose that question, you're going to be listing these functions, 3 here and 2 for kinins, with a few details kind of fleshed in there, but not the diagram. Okay, let's review. All the way back to your notes on Chapter 8 and DNA replication. As usual, bringing a scan file next time and a pencil. I don't want to record it up here at all, it's fine. This does right here. Extra. The real point is we want B. We're going to have 4 multiple choice and 2 discussion questions next time. Now, the process called DNA replication. The unwinding always begins in the middle of the molecule. The nucleotides for copying are called DNA nucleotides. DNA. This process copies both strands of the whole molecule. On average, for every 10 billion new nucleotides put in, 1 has the wrong base. Only 1 in 10 billion wrong number is common. This process has self-correction, I sometimes call pinpoint correction. Enzymes cut out and fix 1 nucleotide as well. This process does not have do-overs. Now the process called transcription. The definition is the cell takes DNA and makes RNA. Here the unwinding always takes place wherever the gene is we're working with. The nucleotides for copying are called RNA nucleotides. This process copies only 1 strand of 1 gene. On average, for every 100,000 nucleotides put in, 1 has the wrong base. Mistakes are more common here in the other process. And transcription has no self or pinpoint correction. It doesn't have that. Thank goodness it does have do-overs. The cell messes up here, it's going to degrade all the RNA and just starts over the whole process and tries again. Now for translation, we have some fairly fancy diagrams, but know just what I mentioned about translation. The definition is the cell takes RNA and makes protein. This involves M or messenger RNA. This is the instructions or the message to make a protein. So a codon is a group of 3 bases in mRNA. Connect mRNA and codon. And a codon is a code for 1 particular amino acid. Now tRNA is transfer RNA. Now Transfer RNA takes amino acids from the cytoplasm to the ribosome. Puts them in the right place. The main three bases in tRNA is called the anticodon. Connect tRNA and anticodon. Now, AMP binds to and activates amino acids. Connect AMP and amino acids. Now, release factor is a protein that binds to a stop codon to stop the process. As in, we're done releasing proteins. So just focus on those highlights that I mentioned. Now the bacterial transfer of DNA. You should know a definition for conjugation, sometimes called bacterial conjugation. This is the transfer of a plasmid from one bacterium to another. You can't memorize the definition or memorize what? Conjugation, plasmid. Focus on key terms that go together. Now, transformation. Transformation. A bacterium acquires new characteristics because it absorbed DNA fragments from another bacterium that recently died. I connect transformation, I guess, from died. Now, many years ago, a man named Griffith tried to find a vaccine for Streptococcus pneumoniae. A vaccine for that. He never did. But he knew something right away. He had two strains. One made rough collies, called the R strain. One made smooth, S strain. He knew pretty quickly in his research that the R strain he called non-pathogenic. The S strain was very pathogenic. Now, don't worry about all of his trials, just the one big breakthrough. When he put RL plus SD in the same lab rat, his prediction was the lab rat should be fine because R is alive, is also non-pathogenic. S is very pathogenic, is also dead in that case. To his great surprise, that lab rat died on him. He was very surprised. He discovered then the process called transformation. Transformation, discovered that. He couldn't figure out, though, what are they picking up the dead ones transform. Years later, Avery figured that out. Now again, don't worry about all of his trials. No, the one Avery trial is a big breakthrough. RL plus SD minus DNA. He destroyed all the DNA of the dead bacteria. Finally, the lab rat lived. So Avery said this: DNA does transformation. DNA gives cells characteristics. DNA gives us characteristics. DNA is the genetic material. We proved that. Now for transduction there, no, just the definition. This is the transfer of DNA from one bacterium to another by way of a bacteriophage, which is a virus. I connect, if I was you, transduction and bacteriophage. Try to visualize which two terms go together. Now chapter 14. Now let's take a look at the first possible discussion question. So find your notes on normal flora. If I choose this one, I want a definition. These are microorganisms that can live and reproduce in our bodies, usually without causing harm. Also mention this. The big advantage of having this is they compete with pathogens for nutrients and space. Then mention for me 3 situations where normal flora microbes can be harmful. They're usually not, but they can be. The first situation is that they get somewhere in the body not supposed to be. I want you to mention both examples for me. E. coli in the bladder is a problem. Candida albicans in the blood is a problem. The second situation, they reproduce too much and there are just too many of them. Too many. As in, way too many Candida albicans in our mouth is called thrush. Way too many. The third situation is if they can take advantage of our compromised or weakened immune system, such as Staphylococcus aureus, pneumonia on top of the flu. Now, when you write out things like Staphylococcus aureus, if your spelling is close, I don't count off the spelling. If it's way off, it could be a point. So look for those things if I choose that question. The definition, the big advantage in those three situations. They might be harmful. Now let's take a look at multiple choice up top there. And it says inapparent. Now I have left off quite a few terms here. But find your notes on inapparent. Here, the symptoms are so mild. So mild. You can be unaware you have infection. You can be unaware. Now connect these two terms for sure, inapparent and chlamydia. If a patient has chlamydia and they don't get treatment, it may cause things like sterility in females. If the infection is called latent is inactive. Such as the herpes viruses, connect latent with herpes. For example, herpes simplex 1 causes cold sores in the mouth. The virus will go to nerves. If they reactivate later, the cold sores reoccur. Genital herpes simplex 2 causes genital herpes. They can again reoccur from nerves. Varicella zoster causes chickenpox. If they reactivate the nerve later in life, that'd be shingles. Now take a look at the second discussion question at the bottom. You're going to talk about these three terms. I'll give you, kind of prompt you to talk about these three terms. A reservoir is just something that allows a pathogen to stay infectious and reproduce. The main example here is warm-blooded animals. Although food and water are sometimes reservoirs, a vector is an insect that transmits the pathogen from host to host. Now at that point, I want you to mention two examples. For malaria, the reservoir is humans. The vector is mosquitoes. For Lyme disease, the reservoir is field mice, the vector is ticks. Then, if you're a carrier, you have no symptoms, but you're infectious. And I want you to mention this. You can be infectious before your symptoms start. It's called incubation period. You can be infectious after your symptoms are over for a while. That's called convalescence period. Mention those things. We'll skip illness. That's pretty obvious. You're infectious when you're really sick. Mention incubation and convalescence. They're tricky because you have no symptoms, but you're infectious somewhat. Now back to multiple choice and find where it says contact, vehicle, and vector. I call these the three specific categories of disease transmission. Now, if we say direct contact, we're going to skip the definition there, but to know the example, I like that example. So, warts and genital herpes, direct contact. Now, indirect contact could be shaking hands, like common cold and flu. Indirect contact could also be the sharing of needles. Picking up HIV, hepatitis B and C. Now, do know a definition for droplet contact. Here, the pathogen can be infectious in droplet for 1 meter or less. 1 meter or less. So, the flu, whooping cough, whatever. Now, if the vehicle is food, that could be botulism. If it's water, that could be cholera. If it's body fluids, that could be HIV, hepatitis B and C. Now, know a definition for vehicle air. Now, the pathogen can be infectious in a droplet from much farther than 1 meter. Now, it's clear across the room, like measles and tuberculosis. A vector is an insect. So a mechanical vector is not an insect bite. That's like the flies feed onto your food. You can pick up bacillary dysentery by mechanical vector. A biological vector is an insect bite, like malaria, mosquito bite, or Lyme disease, tick bite. You may see a question like this next time. What category of transmission is cholera? The answer is vehicle, water. How about tuberculosis? Vehicle, air. How about bacillary dysentery? Vector, mechanical. So, next question like that. Now find your notes on nosocomial infections. These are hospital-acquired infections. Every year in our country, about 2 million patients get these each year. Of those, about 20,000 die from them. A very common cause of these is MRSA. This strain of Staph aureus is hard to treat. Also E. coli and Clostridium difficile. Now, chapter 15, we have a few questions here about bacteria. The first question in chapter 15 was, how can bacteria get in our system in the first place? The so-called portals of entry. Which of know these three in order of prevalence? By far most common is mucous membranes, followed by the parenteral route of breaking the skin, followed by the skin route. There'll be bacteria infecting the open hair follicles or sweat glands. Now, under mucous membranes, note some examples here. The most common example of these membranes is respiratory tract. Bacteria going that way, you might get things like tuberculosis, bronchitis, pneumonia. Under mucous membranes still, we have the gastrointestinal tract. If you swallow bacteria here, you might get botulism, cholera, typhoid fever. Then the genitourinary tract, the urethra, or female vagina also. But that'd be things like chlamydia, gonorrhea, and syphilis. Under parenteral route, note two examples, that'd be tetanus and gas gangrene. They're picked up by breaking the skin. Under skin route, the bacteria infect the hair follicle like a pimple or a boil. How can bacteria stay in our system? Well, they have, unfortunately for us, adhesins. These are certain proteins on bacteria that bind to receptors on our cells as a way of staying in our body. Know 5 places bacteria might have adhesins: on their cell wall, on their capsule, their flagella, their fimbriae, and the pilus. How can bacteria avoid our natural defenses? Well, the M protein is on Streptococcus pyogenes. The M protein has 2 functions. It acts as an adhesin. It binds to receptors on the cells that line our throat. This is strep throat. It also prevents or blocks phagocytosis. Now, a bacterium called Mycobacterium tuberculosis has a strained cell wall with a bunch of lipid called myconic acid. Because of that strained cell wall, it survives and reproduces inside our macrophages. With tuberculosis, traditional phagocytosis doesn't work. The last question for this chapter, how can bacteria actually make us sick? It's toxin. The two kinds in general are exotoxins and endotoxins. Now, think of an exotoxin as a liquid waste product made of protein and given off by some of the Gram-positive bacteria. There are three types of exotoxins, beginning with cytotoxins. These here either damage or kill cells, as in our cells. So Corynebacterium diphtheria gives off a cytotoxin that prevents our cells from making protein. This will kill some of our cells, meaning perhaps organ damage, organ failure. Streptococcus pyogenes. That's a cytotoxin that will damage our blood capillaries. That's a condition called scarlet fever. Now, neurotoxins block nerve impulses. Clostridium botulinum gives off a dangerous neurotoxin that blocks the first nerve impulse to muscle. That means muscle paralysis. Clostridium tetani gives off a neurotoxin that blocks the second nerve impulse to muscle. So tetanus is constant muscle contraction. Now, entero means intestine. So these mess up the cells that line the intestine and be diarrhea. So Vibrio cholera is often enterotoxin, makes the cells that line the intestine lose their water. It's very watery diarrhea. The other main kind of toxin is called the endotoxins. They're a component of the cell wall. For some Gram-negative bacteria, they're made of lipopolysaccharide, which is fat and sugar. Our example is Salmonella typhi. If this winds up in your bloodstream, then you have typhoid fever. Now, a macrophage will ingest the bacterium like it's supposed to, and also digest like it's supposed to. But now the endotoxin by itself will stimulate the macrophage to release IL-1. What do you connect with IL-1? Fever. That's a big fever. But it also makes the macrophage give off TNF, which causes shock. It's going to destroy good capillaries there and cause shock. So IL-1, fever. TNF, shock. Now from today, chapter 16. Our first line of the National Defense Against Infection, mechanical and chemical barriers. A good mechanical barrier is the skin is intact. Also, mucous membranes like nasal cavity, good mechanical barrier. A good chemical barrier is stomach acid, as well as lysozyme. Lysozyme is an enzyme. It dissolves peptidoglycan and kills really well Gram-positive bacteria. There are three places we all have lysozyme. Some, white blood cells, perspiration, and tears. Now, the second line I call PIC, beginning with phagocytosis. You sure know what the three white blood cells that do this actually engulf. Neutrophils engulf bacteria only. A macrophage engulfs bacteria and viruses. Eosinophils engulf the entire antigen-antibody complex. Know the highlights of the four steps, the highlights. You may see a question like this. In step 1, chemotaxis. What exactly are neutrophils attracted to? Activated complement proteins, activated kinase. As they move towards them, they're also moving towards the bacterium. Step 2 is called attachment. I can ask you, what can block step 2? Well, a capsule and proteins. Step 3 is called ingestion. They use pseudopods to ingest. Connect ingestion and pseudopods. Now, then connect step 4, digestion, with lysosomes, with the enzymes there. I sometimes ask this question, what compartment is used to expel the waste products, that's residual body? So, the lysosomes are bags of enzymes. The residual body expels the waste products out of the cell. Now the I of the pig, which is inflammation. The two main examples would be the area around the cut turning red, extra blood there, and stuffy nose. In the nose, you have extra mucus sometimes. Why do we bother and do inflammation? There are 3 reasons. We can find the pathogen, destroy the pathogen, repair damage. Now take a look at the last possible discussion question, right bottom. If I choose that one, you're going to write about 3 functions of complement proteins. And if you want to, you can abbreviate and say, you're helping out with phagocytosis by doing opsonization. And don't really draw this, just say it this way. This right here means they coat bacteria to attract things like neutrophils. Here they're coating bacteria to attract things like neutrophils. I wouldn't draw that, I would say it that way. Also, they help inflammation by stimulating mast cells and basophils. Mast cells and basophils. Enough of that function. Then they help out sometimes the immune system by doing cytolysis. Now, right here, I want to mention this. Here the complement proteins work with antibodies and make bacteria burst. Cytolysis. Here, complement proteins work with antibodies and make bacteria burst. So I don't want these diagrams. I want these 3 functions with a bit of explanation, like on 1 and 3, especially 1 and 3. Then you got to write down the 2 functions of kinins. I might say it this way. Kinins help phagocytosis by again doing opsonization. Now, under kinins, I would just put this, because the explanation about coding, you already did that. So put this under kinins for phagocytosis to abbreviate that. Then, how can the kinins help out with inflammation? By causing to happen both vasodilation and capillary leakage. Mention those 2. That's how kinins help inflammation. So I'm wanting some lists there of explanation and not so much diagrams, if I choose that. If you have questions, I'll be around. Otherwise, that's all. Don't forget your recorder.